While pop culture explores fictional fungal outbreaks in humans, the reality is a 48-million-year-old pathogen that has been executing complex mind control in tropical rainforests since the Eocene. The victim is the carpenter ant, Camponotus leonardi. These insects live high in the canopy, navigating a safe network of aerial trails to avoid the hazards of the forest floor. They are highly specialized for this arboreal existence, remaining in the canopy to stay away from ground-dwelling predators and pathogens. However, gaps in the canopy occasionally force the ants to descend. On the forest floor, a microscopic Ophiocordyceps unilateralis spore attaches to the ant's exoskeleton. The ant returns to its colony unaware of the infection, but a biological countdown has begun. The spore uses mechanical pressure and enzymes like chitinase to dissolve the ant's armor and drill inside. The fungus multiplies into yeast-like cells, forming a dense network around the brain. It then releases mind-altering metabolites, such as sphingosine, interfering with neural pathways. This biochemical interference manifests as severe full-body convulsions, eventually causing the ant to lose its grip and fall from the canopy. The parasite now exerts direct control over the host's locomotion, navigating the ant away from its colony toward its final destination. This diagram shows a vertical scale of the forest floor. The fungus forces the ant to climb exactly 25 centimeters up a plant stem, reaching a microclimate with the specific temperature and humidity required for the fungus to grow. At exactly solar noon, the fungus triggers a final behavior. The ant bites down on a leaf vein with abnormal, permanent force. To lock the host in place, the fungus secretes chemicals that strip the ant's mandibular muscles of leucine and energy-producing mitochondria. The muscles atrophy and seize, creating a permanent lockjaw. Over the next 4 to 10 days, the fungus consumes the internal tissues and erupts a fruiting body directly from the ant's head. From this elevated position, the fungus releases spores onto the forest floor, creating high-density graveyards of infected ants. The ants counter this through social grooming, while a fungal hyperparasite frequently attacks the zombie fungus itself, limiting its ability to spread. This survival strategy has remained consistent for 48 million years, with every behavioral shift and chemical secretion driven by the singular goal of reproduction. Biologists view this as an extended phenotype, a process where the fungus's genes express themselves by manipulating the physical actions of a different species. The metabolic toolkit used to hijack the ant's brain also serves a defensive purpose, producing compounds that protect the host carcass from decay and scavengers. In the lab, researchers have found that these secondary metabolites are heavily bioactive in humans, meaning they can trigger specific biological responses in our own cells. Scientists are specifically isolating naphthoquinone derivatives and polyketides. This diagram shows how these structures are being categorized for their anti-malarial, anti-cancer, and antibacterial properties. By deciphering the chemistry of this 48-million-year-old parasite, researchers are identifying new ways to turn evolutionary weapons into life-saving human medicine.